So I have noticed that we are live. I would like to welcome again all participants of the second poster session at Tropentag 2020. We are live and stream on YouTube as well. My name is Jan Banota and I'm a professor at the Faculty of Tropical Agri-Sciences at Czech University of Life Sciences Prague. And it is my great pleasure here to be as a chairman with all of you during this virtual conference. So we are going to start with the first presentation. This presentation is by Mr. Ayatingbo, which is about the drying kinetics and characterization of dried osmotically pre-treated wheat flesh and yellow flesh cassava. Yeah. So please, if we can listen the first presentation or the video of the first presentation. Thank you very much.
Hello. Can you hear me?
Yes, we can, but we are going to hear the video. Did you upload the video?
Yeah, I uploaded the video and I'm really opening it right now on Zoom.
Yeah, but I think you are going not to share your presentation with us, or they are going to play us the video from the WhatsApp, if I'm right. Okay, so you can stop the sharing of your screen.
Okay.
If I can ask.
I stopped it. Okay.
Good.
So are you playing it from your side, or do I have to open it?
No, we are going to play it now.
Okay, then. Thank you.
So then we are going to ask you the question. We can start right now. Great.
Okay, thank you. Good day, everyone. My name is Oluwatoyin Ayetibo. The topic of this poster is drying kinetics and characterization of dried osmotically pretreated cassava. I work with Dr. Sajid Latif, Dr. Abbas Adebayo, and Professor Joachim Mueller. Cassava is also known as manioc or yucca, but is highly susceptible to post-harvest physiological deterioration. Therefore, drying was considered as a means to arrest this problem. Osmotic dehydration was considered because it is simple, cost-effective, and requires low energy input. Drying parameters such as moisture ratio and drying rate and diffusivity are important in understanding the behavior of materials during drying. And an optimization of drying conditions can also be conducted using response surface methodology. The effect of these drying conditions on the physicochemical properties such as total carotenoid content, total cyanogenic potential, and microstructure of cassava can be studied. To do this, we used two varieties of cassava, the white and the yellow varieties, which were cut into regular sizes and were dehydrated for 5 hours at 45 degrees centigrade in a 70-degree brick salt sugar solution. Afterwards, they were dried at different temperatures of 50, 65, and 80 degrees centigrade and different air velocities of 0.5, 0.9, and 1.3 meter per second. An optimization of these drying parameters or conditions were carried out using a 3-level factorial experimental design, considering the temperature and air velocity as the variables and responses as drying energy consumption, shrinkage, drying energy, drying time, and diffusivity. Moisture ratio was modeled by 4 mathematical models, Page, logarithmic, Newton, and Anderson-Parbis models, while drying rate was modeled by Peleg, Page, and Anderson-Parbis models. The microstructure was determined by scanning electron microscopy, shrinkage by changes in the volume of the cassava cubes, total cyanogenic potential by pre-crate paper method, and total carotenoids by spectrophotometry quantification. From the results, we see that the optimal conditions for drying cassava was found out to be at 80 degrees centigrade at 0.5 meter per second air velocity, and we saw that the predicted, validated, and experimental data were very closely related to one another because the response models were significant and with an insignificant lack of fit. Therefore, we can confidently predict the drying parameters using response surface methodology. It was found that moisture ratio was best fit to the Newton model, It decreased with increasing temperature, but air velocity had no effect. Drying rate was fitted best to the Peleg model, and it occurred only in one falling rate. Drying rate increased with temperature and air velocity, just like diffusivity. Osmotic dehydration and drying reduced the shrinkage of cassava by about 50%, and this is an advantage. And this shrinkage increased with temperature and air velocity. Total cyanogenic potential also reduced by about 50 to 60%, which is also advantageous, making the cassava safer for consumption. Microstructure showed that the starch granules were lost or solubilized, and the cell wall structure was ruptured. However, the downside of these processes is that total carotenoid content was reduced to about 15 to 36% of initial carotenoid content. In conclusion, we see that drying conditions influence the drying kinetics and physicochemical properties of cassava varieties. The optimal conditions for drying osmotically dehydrated cassava was at 80 degrees centigrade and 0.5 meter per second. And the osmotic dehydration process has some advantages such as reduction of shrinkage and total cyanogenic potential. Thank you.
Okay, thank you, Matthew, for your virtual presentation. And now we have place or time for one question. Yes, Iris, please.
Hi, Toyin. Thank you for your very nice presentation. I have just a question. It's not really a question, just to be clear. I mean, what kind of specific humidity or relative humidity did you use during your experimentation? And whether you use the through flow or overflow mode in this precision dryer? As I know that we, in our institute, were able to do multiple experiments and different methods. Thank you.
Yeah, thank you, Iris. For this work, the specific humidity that was considered was 10 grams per kilogram dry air. And this is because it has been found that under the drying conditions in this part of the world, the relative humidity is comparably lower compared to that in Africa. We could have used 25 grams per kilogram, but then it's really going to take much more longer to dry, and there might even be mold development during drying. Secondly, your second question was about if we used overflow or true flow, and we used true flow overflow drying in this work, because if we use true flow, after drying, the density of the cassava cubes may be reduced, and this may change the orientation of the cassava cubes during drying, and this would affect our drying kinetics. Thank you.
Okay, thank you very much. So let's go for another poster presentation by Oladi Adergibe.
Yes.
Aderibi.
My name is Olaide Aderibi.
Yeah, you are here. Great. So don't worry, we are going to listen your presentation, which is uploaded. So the nutrient and anti-nutrient composition of biotin cubes developed from fermented commodities of Ricinus communis. Yes, please.
Olaide Aderibi. I'll be presenting a research work titled Nutrients and Anti-Nutrient Composition of Bouillon Cubes, developed from fermented condiments of Ricinus communis seeds. Fermented condiments are part of the diet in Africa. They are usually in slurry, paste, or loose solids form, and they are wrapped in leaves. Ricinus communis happen to be one of the seeds used for preparing this condiment. It is known as castor oil seeds or castor oil bean. This work seeks to develop bouillon cubes from the condiments so as to remove some of the safety concerns and to also improve the attractiveness of the product. We follow the normal traditional fermentation procedure. After preparing the condiments, they were mixed with cassava starch as a binder. They were then compressed and shaped into cubes and then dried. Condiments and binder were mixed at ratio 20:5, 20:10, and 20:20 and subjected to chemical analysis following standard procedures. The graph here shows the proximate parameters of the condiments. As the proportion of binder, which is cassava starch, increase, protein and fat decrease, while carbohydrates increase. This is of interest because these condiments are known to be protein sources, and the addition of binder has reduced the protein composition. The table beside it shows the phytates and mineral composition. Phytates increased as binder increased while minerals increased. Phytate is an antinutrient, and it was the only antinutrient that showed significant differences. Vitamin C did not show any significant difference between all the three condiments. In conclusion, production of fermented bouillon cubes of Ricinus communis is promising. However, the cubes produced have decreased protein content and increased phytate content. We hereby recommend that the use of high protein binders or a blend of binders that can retain the protein composition should be considered in the development of these novel products, as they are known to be rich sources of protein. Thank you.
Yeah, thanks for the virtual presentation. All ID is here with us, so the question is possible.
I have a question.
I can't
Yes,
hear
please.
anything.
I have a question
I
for
didn't even
Lady.
hear my presentation.
Can you hear? Can you hear me? Hello?
Hello, All ID, please, can you hear us?
Hello? I don't
Hello.
think.
It seems that.
Yeah, I don't think she hears us.
We lost her, maybe. All ID, can you hear us? No. So no questions. I'm sorry. It seems that the connection is bad, so that's the problem with those virtual meetings. Maybe she will be online later at the end, we can ask. So let's proceed for the other presentation, which is a presentation of Filip Wojacek, and the title is Effects of Heating on Fatty Acid Composition of Edible Oils and Fats. And I would like to ask Olaidi to mute herself.
Hello. Hello, my name is Filip Wojacek, and I would like to introduce to you my research about effect of heating on fatty acid composition of edible oils and fat. Main objective of this study was to investigate the effect of long-term heating on fatty acid composition of 7 types of oils and 1 type of fat at high temperature. And minor objective was to compare the thermal oxidative stability of sunflower oil against the high oleic sunflower oil. For analyze 7 types of oil, such as soybean oil, coconut oil, palm oil, sunflower oil, high oleic sunflower oil, rice bran oil, and rapeseed oil, and 1 fat hog lard were used. Samples were heated one by one in conventional fryer for 24 hours at 190 degrees of Celsius. Base catalyzed transesterification catalyzed by sodium methoxide was performed on each sample, and in order to determine the composition of fatty acid methyl esters, the gas chromatography mass spectrometry analysis was performed. In total, I detect 27 different types of fatty acids, and after 24 hours of heating, all tested oils and fat became more saturated. The changing in saturation you can graphically see in figure 5, and the exact changes in the content of each fatty acids in each of oils and fat you can find in tables in my study. On these chromatographic peaks, we can see how the decreasing trend in content of linoleic acid influence the content of oleic acid in coconut oil because of degradation of double bonds. In conclusion, the heating caused degradation of fatty acid double bonds, and according to my research, the best thermal oxidative stability has palm oil, followed by rapeseed oil, and the least stable fatty acid composition has coconut oil and rice bran oil. High oleic sunflower oil has better thermal oxidative stability than the sunflower oil, and it seems that the oils which contain fewer types of fatty acids are more stable than oils rich in different fatty acid species, but this statement needs further research. Thank you for your attention and have a nice day.
Okay, thank you, Philip, for your virtual presentation. I have the question, if there are any questions to Philip.
Yes, I have a question, Phillips.
Yeah.
Thank you for your presentation. The question is that what do you think could be responsible for the palm oil having a lower thermal oxidation capacity?
Lower thermal oxidation capacity is because it contains higher amount of saturated fatty acids, and therefore is this oil often used for frying or was used because now it's in last year it's very discutable about the healthy benefits of this oil, but the stability is because of high content of saturated fatty acids in this oil.
Yeah. Do you think it might also be due to the presence of relatively high amounts of antioxidants, like maybe vitamin E? You have some tocopherols in the oil.
I didn't study it, this content, but I think that yes.
Okay, it's just an observation. Yeah.
Okay, thank you very much for this contribution. And let's go to hear another poster, which is going to be presented by Iris Ramay, and it's about the influence of self-compaction on the airflow resistance of grain bulks.
Dear auditors, my name is Iris Ramay, and I'm doctoral candidate at University of Hohenheim Institute of Agriculture Engineering. I would like to welcome you to my poster entitled Influence of Self-Compaction on the Airflow Resistance of Grain Bulks, which was within the frame of a project focused on the aeration of stored grains with refrigerated air. Nowadays, these practices are among the most effective non-chemical methods in use, which apply conditioned air through the grain bulk to guarantee quality preservation and safe storage. Nevertheless, some constraints have been encountered in estimating the airflow resistance of grain bulk, which has led to aeration deficiencies. Hence, the objective of this study was to assess the influence of self-compaction on the airflow resistance of grain bulks. As an experimental basis, a testing apparatus filled with wheat grains was employed. A coherent set of airflow velocities ranging from 0.01 to 0.15 meters per second and storage times ranging from 1 to 236 hours at 4 grain depth levels were applied accordingly. The relationship between pressure drop and airflow velocity was assessed experimentally and modeled theoretically by the model proposed by Lee. The results showed that the moisture content of the grain bulk did not change during the experimentation, and no supplementary drying took place. It was further observed that the airflow velocity had a substantial impact on the pressure drop, since pressure required to force air through the grain bulk was dissipated continuously due to friction and turbulence. Furthermore, results demonstrated an increase in the pressure drop throughout the depth of the grain bulk. This behavior was ascribed to the self-compaction arising from the Böder stresses imposed by the dead weight of the bulk. The increase in the grain depth resulted in higher stresses due to higher imposing weight. The resultant stresses are believed to initially influence the reorientation of the particles and afterwards cause their plastic deformation once the rupture force is achieved, which eventually decrease the void spaces or porosity, increase the bulk density, hence increasing their airflow resistance throughout the grain depth. Similarly, the storage time demonstrated a significant effect on the pressure drop. This is ascribed to the temporal rearrangements and deformations of the kernels until the grain bulk was completely settled. The influence of storage time, grain depth, and airflow velocity on the pressure drop is graphically presented in Figure 2, whereas the porosity and bulk density variations imposed by self-compaction are displayed in Figure 3. To compensate for the larger pressure drops, extra power supply for aeration are prerequisites to overcome the resistance caused by self-compaction, hence resulting in a change of the pre-designed operating point of the fan. It can be concluded that during the storage phase, grains are exposed to burden pressures that contribute to self-compaction. This phenomenon affects considerably the ventilation of grain bulks and thereby would be considered in the planning and design of aeration systems. I would like to thank you very much for your attention, and I would be looking forward to your questions.
Sorry, you are muted. Sorry.
So I am
Yeah.
muted, and
Yeah.
now I'm with you. Thank you, Iris, again for your virtual presentations. We have a time for questions.
I have a question for Iris,
Yes.
and it's, do you think the walls of your compartment in which the grains are studied, do you think they might have some kind of effect on the compaction of the grain particles themselves?
Thank you, Twin. Yes, I think it can be because we use this circular bin containers or silos, which are most often used also in practice. So if we would have something like rectangular, maybe in the edges, that can be some lack of grains, or can create some spots in which grains are not properly located. So I think can help, or can influence, but I'm not sure how and in which extent, so that would need further investigation.
Okay, thank you.
Welcome.
I have one also short question. Did you investigate the effect of the compaction with time?
Yes, we did. This was the outcome of the result, that we have a certain self-compaction over the grain depth, but also over time. Yes.
So it is affected by time as well.
Yes.
Okay. Good, thank you very much. So let's go to proceed for another poster presentation by Joshua over there, and it's about the response surface methodology models for optimization of traditional fermentation of coffee leaves.
Hello everybody, my name is Joshua Mbakawade from the University of Nairobi, Kenya. And on behalf of my other co-authors, I am presenting on response surface methodology models for optimization of traditional fermentation of cowpea leaves. The work was undertaken under the Fruits and Vegetables for All Seasons project funded by the BLE. Cowpea leaves, these are vegetables that have been vastly utilized in sub-Saharan Africa, especially Western and Eastern Africa. And the communities in the utilization of this vegetable do employ diversified processing techniques, one of them being spontaneous fermentation. However, in the case of spontaneous fermentation, the process tends to be slow and the product quality do vary. This study therefore sought to optimize the fermentative parameters for production of the traditional soured cowpea leaves. The approach that was used in this particular study, cowpea leaves were harvested at the optimal maturity stage, 36 weeks after emergence, and subjected to spontaneous fermentation using low-cost processing techniques. The experimental design, the response surface methodology models in the design expert software were used to generate 20 experimental runs, with the response variables being pH and table acidity, whereas the fermentative parameters under investigation included concentrations of salt and sugar and the period of fermentation. Statistical analysis was effected using an overflow quadratic model. In the results, the models were found to be significant, with them accounting for 89% variation in the pH and 60% variation in the table acidity. As you can see illustrated in the graph, as the sugar concentration was increasing, the pH decreased. Whereas on the other hand, as the sugar concentration was increasing, the terrible acidity increased. Salt concentration did not significantly affect either pH nor the terrible acidity. Salt concentration, sugar concentration, and period of fermentation, these are factors that independently have been established to influence the rate of fermentation. This is because they do improve the microbial activity. However, when these factors are combined, the increase in the microbial activity do go up to a point where the osmotic stress do set in, limiting further increase in the microbial activity. The fermentative parameters were determined as salt concentration of 2%, sugar concentration of 5%, with 16 days of storage. Whereas the response parameters were established with a pH of 3.8 and terrible acidity of 1.2% as the optimal points for fermentation. Therefore, in conclusion, in averting the challenges of poor product quality and salt fermentation process that is rampant among the traditional communities, it would require the addition of sugar and salt to the vegetable, and also the optimization of fermentation period to 16 days, which has been determined in this particular study. Thank you, and I'm looking forward to your questions.
Okay, thanks again for the virtual presentation. It was quite fast from the beginning, but it was regular at the end. So good. So we have Joshua here with us, so we can ask the question.
Yeah.
Yeah.
So Joshua, thank you for the presentation. I just want to ask with regards to, because this question was also asked when I worked on osmotic dehydration of cassava. I was asked by some interesting question that we are trying to reduce calorie or sugar intake and salt intake. So why are we osmotically treating our materials? So I pose this also as a challenge to you. Why do you think adding salt and sugar to vegetables, which if it was ordinarily consumed, will not contribute to any health challenges? Or
Sorry, my internet had a challenge. Can you just come again with the question?
Okay.
It's okay.
Just very shortly. In nutrition, most times we are trying to reduce consumption of sugar and salt because of the health risks associated with these materials. So why do you think this could be attractive to processors or consumers?
Okay. I've gotten your question. Thank you for that particular question. With for food processing, the process that we are using in the fermentation, the sugar that is added and the salt that is added are simply just to ensure that the process is a bit optimal and more improved in terms of the product quality. In real sense, we cannot say that we necessarily need to do away with the sugar nor the salt, because actually the salt is actually necessary also for the taste and such like things. But the sugar will actually be used and be converted into lactic acid. Therefore, it means actually eventually it will not be a factor in terms of consumption, in terms of ruining the nutritional quality of the particular product. But this particular study actually sought to improve both the health product, the health benefits derivative from this particular product, and also went a long way in terms of ensuring also the quality of the product that comes out is a little bit more optimal. So it is not much of a concern, especially when we are going towards the fermented products, because the sugar will be utilized, but the salt is actually really minimal. Yeah.
Okay, thank you.
Good, thank you very much. The another presentations by Mr. Ann Miano is going to be skipped because we don't have this video, and we're going to proceed directly to the last one by Huyen Chao Dang. It's about the valorization of bioproducts from coffee processing to improve the sustainability of the coffee value chain in Vietnam.
Good evening, everyone. My name is Huỳnh Châu Đặng from Vietnam. Currently, I am doing my PhD thesis at Leibniz Institute of Agricultural Technology and Bioeconomy in Potsdam, Germany. Today, I'm gonna introduce you the coffee jar project that I and my research group are working on. This project is funded by German Federal Ministry of Food and Agriculture. The goal of this project is to improve the sustainability of the coffee value chain in Vietnam. One of the main research questions is, can hydrothermal carbonization improve the combustion characteristic of the coffee byproducts from the wet coffee berry processing? The wet coffee berry processing produces higher quality coffee beans than the dry processing, but also a large amount of wet solid waste, and the wastewater treatment plants are often overloaded. We are studying how to use hydrothermal carbonization to improve the heating value of the byproducts and the pelletizing process to produce hydrochar pellets that will be used in the processing plant as fuel to dry the green coffee beans. The preliminary results here on the poster show the low heating value of the coffee byproducts as well as the heating value of spent coffee grounds, the byproduct left over after making a cup of coffee. Currently, due to the COVID pandemic and restriction in travel, hydrothermal carbonization experiments with coffee berry byproducts must be postponed. We have made a few hydrothermal carbonization runs and pelletizing tests with spent coffee grounds. The final hydrochar pellets have a better heating value and lower ash content in comparison to wood pellets and the initial feedstock. The outcome of the project will be an integrated process design that will treat the byproduct, reduce the volume of waste, and increase the heating value. This solution can support farmers and producers to improve the sustainability of the coffee value chain in Vietnam and also provides a basis for adoption of hydrothermal carbonization application to other coffee production regions. That is my poster. Thank you so much for your attention.
Thank you very much. Mrs. Huyen Chow Dang is here with us, so we can ask questions.
Can I ask a
Yes.
question?
Yeah.
Thank you so much, Cheldon, for your presentation and that great particular concept coming across. Now, because you are talking of sustainability with, I know your country is also signatory to the agreements, especially the ones on climate change. I don't know how this process, if you are looking also to check the carbon footprint, and then in the long term, do you see it as something sustainable? Because I have seen the kind of combustion taking place. Would it be something that in future there are also issues to do with the climate change? Thank you so much.
Thank you so much for your questions. Actually, like in the project, we are not working the CO2 footprint. And for the climate change, so like right now, we just focus on producing the alternative fuel. That's we can replace the fossil coal that the factory are using now in Vietnam. And we, for the climate change adaptation, so we also think about, how can I say, do more research on the CO2 emissions when using the renewable fuel for drying coffee beans. But we did not like have planned to do it so far, and I think that is also a good suggestion. We should have some further research on this to, I can say, encourage or to prove that our product can relay the fossil coal for getting energy. Yep. So I don't know if my answer can can answer your question or not.
Okay, so maybe I have one more about what do you think about the energy inputs to produce such kind of pellets from this kind of bio waste? Did you measure the the energy balance between the input and outputs?
Actually, like we need to do the energy balance and mass balance in the end of this project. But currently, right now, for the hydrothermal the like the energy input, we did not measure it, but we need to heat up the wet biomass to convert it into hydrochar. So we need to heat up the biomass up to 260 degrees in 6 hours. So
Okay.
to turn it into char.
Yeah, so there are some energy inputs.
Yes, exactly. So we need to make the energy balance and mass balance in the end. And yeah, we need to, because the factory, like the coffee factory in Vietnam, we are working with right now. So they are willing to use renewable fuel from the hydrochar, but they want to see the economic and energy benefits. So yeah.
Okay.
Thank you so much.
So one more question is still possible if anyone is interested. I have checked. Yeah. Yes, please.
Thank you, Chao Dang, for your work. I just want to ask that, do you think if inert gases like nitrogen is incorporated during thermocarbonization, do you think there might be possibility of reducing the carbon footprint?
You mean the adding the nitrogens or what?
Yeah.
Right.
Yes, exactly.
Right now we are making 2 runs, one with nitrogen and one without nitrogen, and we will have the analysis later in next month, and we see the results, because yeah, and we also think about that we need to wash the hydrochar after the conversion. Maybe we can wash out the mineral or unwanted compounds, that we have more how can I say, clear char pellets. Yeah, but it's still on the way, because I'm just at the first year of my PhD thesis, so that's just the initial experiment. So yeah, but I think all your suggestions are good for for my thesis. Yeah, thank you so much.
Thank you.
Okay. So if we don't have more questions, I would like to thanks to all of you to participate this session. This was the last contribution. I would like also to thank to the support from the administrative staff from ATSAF for Tropentag 2020. So have a nice evening and enjoy the other sessions of Tropentag still continuing tomorrow. Goodbye.
Bye.
So it's nice to be here with you.